Okay, good morning, everyone. Last time we were here, catalase positive, gram-positive cocci, and we were talking about the epidemiology. Let's repeat it all over again. If you notice here, the S aureus, which is in red, which is the most important out of all these in this chapter, the habitat is normal microbiota. And we said that last time that Staph aureus is the most pathogenic of all in this group. Staph aureus. Epidermidis not very much, Saprophyticus not very much, Micrococcus not at all. But still, some people they have it as microbiota, which is part of the medical team. Anterior nares, nasopharynx, perianal area, skin, and mucosa. These are the areas where we expect to find S. aureus, whether pathogenic or non-pathogenic. Some people, they have it as endogenous strain, although it's pathogenic, endogenous, part of their body. Or you can get it after a trauma, car accident, or somehow a hit by something, or any accident, or by a direct contact. You can shake hands with somebody, person to person, or you can hug anybody, your mother, your father, your sister, your friend. Also indirect contact, aerosolized by aerosols. Sneezing and talking, you can just airborne. Or through fomites. What are the fomites? Fomites, things that we use. They are not viable. Like at home, we use towels. We use toothbrush. In the hospital, we use needles. We use any instrument, medical or surgical instrument, forceps, scalpels, and so on. These fomites also they can transmit bacteria, of which Staph aureus is part of it. Then we have this S. epidermidis. S. epidermidis, yes, it's purely normal microbiota. It can never, in normal conditions, cause any disease, except in a certain very limited circumstances. You can find this on the skin. So if you make culture for any normal human being, you definitely will get S. epidermidis on the skin. In mucosa, yes, yes, you can, of course. Mucous membranes also, you can find them, but mainly in epidermidis on the skin. Endogenous strain, it's part of our microbiota, so it comes originally from us. Endogenous means within. Implantation, usually is epidermis on the surface of the skin, but if you make implantation of teeth inside your jaw, or let's say implantation of kidney with S. aureus, or let's say you make implantation of artificial limb, which is contaminated by S. aureus, it can cause a problem. If by one way or another, this epidermidis, which is normal, can get into your brain, CSF, it will cause a problem. Yes, this is microbiota, but it depends on where. S. hemolyticus, S. lugdunensis, saprophyticus. Saprophyticus is normal microbiota on skin and mucous membranes, and it's endogenous and may cause, this one is more pathogenic than S. epidermidis. S. epidermidis is lighter. This one may cause sterile UTI, urinary tract infection. Sterile, you don't see lots of white blood cells and these things which we usually see. And this is mainly in sexually active females, young males, and they are sexually active. So micrococcus, like Micrococcus luteus, you will find them on the skin and mucosa, and the oropharynx inside the mouth, and others. So what we are interested in, S. aureus comes first, epidermidis, saprophyticus, and micrococcus. The rest you can just have a look at. Again, this is Staphylococcus aureus, the cluster-shaped golden spheres. Aureus means golden, as we said last time. And this is the most variant species in this group. Pathogenesis. This is S. aureus, 3D as a ball. And each bacteria including S. aureus, they have arms. And these arms, they help the bacteria to invade the host. The bacteria wants to go inside us, but how? There are means. These means, we call them arms. They have an arsenal of arms for this. Let's have a look at their arsenal of arms, which we call them variance factors usually. First, surface proteins. There are proteins on the surface of this bacteria that help the bacteria invade the host, as us, like us humans. First, matrix adhesion factor. Protein A, very important. Clumping factor. Fibronectin binding factor. Collagen binding factor. If you look, these are proteins. Of course, these are not the only proteins on the surface. There are many others, but these are the proteins that were investigated by scientists, and they have proven to be variance factors. Then we have another thing. These bacteria have toxins, and these toxins, they are released. They can be just released as fluid, and they go away from the bacteria and land on the tissue of the host and destroy it, so that the bacteria can make use of it, or they can be connected to the bacteria. Of these toxins, we have hemolysins. As you see here, hemo means blood. So hemo, red blood cell. So hemolysins, these are toxins that lyse the red blood cells, the RBCs, erythrocytes. You have leukocidin, PVL. Leukocidin is one for white blood cell, leukocytes. And also we have exotoxins, alpha, beta, gamma, delta. We'll talk about these ones in the next slide, in not in details, but at least very quick idea. And then we have enterotoxins, exfoliative toxin. All of these are toxins that have only one aim: to destroy the host so the bacteria can multiply and flourish. TSST toxin. Then we have enzymes. So either surface proteins, toxins, and enzymes. Example, coagulase. This enzyme clots plasma by converting fibrinogen into fibrin, which prevents phagocytosis. And this, we made use of it in the lab for the coagulase test. It's a variance factor for the bacteria, but also microbiologists make use of it as a test. And the same applies for catalase, which breaks down H2O2. Catalase is a variance factor, but also in the lab it's a test. Hyaluronidase, another enzyme. Lipase destroys the lipids. Staphylokinase, also this breaks the coagulant. And there is also sometimes, if there is an occlusion inside the vein, in certain circumstances, the patient is given an injection of staphylokinase to break down the coagulum in the veins. So this enzyme, which is originally a variance factor for Staph aureus, people had made use of it to treat as a treatment. So these variance factors, bacteria use as destructive methods to destroy the host, but us humans, microbiologists, we make use of them in the test, and we made use of them as treatment, as in the case of staphylokinase. Penicillinase breaks down the penicillin, which is antibiotic. Nuclease breaks down the DNA. Again, surface proteins, toxins, and enzymes. Let's go to these, not in details, but in a very quick idea. So first, we are going to give you just one line of each variance factor. First, capsule. The capsule is made of polysaccharide, and these capsules they are a layer around the bacteria, and this layer is sticky, slimy, and like a mucus. So it helps two things. First, it sticks on the surface of the host, tissue of the host, because it's sticky, like a mucus, sticks there and makes a biofilm. Secondly, if the macrophage comes and wants to eat and phagocytize this bacteria, this capsule, which is sticky and slimy, it prevents phagocytosis. And thirdly, it inhibits drug penetration. The drug wants to penetrate inside the bacteria to kill the bacteria, and this is like a shield to the bacteria. Sticky, and the drugs cannot go inside. Peptidoglycan of cell wall. You remember in the gram stain last week in the lab, when we said that the cell wall consists of peptidoglycan layer in the gram positive and thin peptidoglycan layer in the gram negative. This one here activates the complement, releases interleukin 1 and chemotactic factor for neutrophils, PMNs, polymorphonuclear cells. They are chemotactic factors. What is chemotactic? They send signals to the white blood cells to come to the site of infection. Activate the complement. Complement, you will have then protein A, a surface protein as we saw earlier, that is on the surface of the bacteria, and it has an affinity for the FC of IgG, immunoglobulin G. So this protein M has an affinity to the FC part of the immunoglobulin IgG to prevent clearance of S. aureus. Then also we have cytotoxins, toxins that are released from the bacteria, and they are released, secreted, and they go away. These secreted toxins, like alpha, beta, gamma, delta, they go to our tissue as host, and they attach to the tissue, get inside, and destroy the cell. So all of these here are valence factors that benefit the bacteria to destroy the host tissue so that it can make a foothold inside our bodies. Toxins, this one also, panten-valentine leukocidin, PVL. This also lyses the neutrophils, the white blood cells, PMNs. It goes inside, see these golden spheres, they go inside the neutrophil, as you see here, and they multiply inside, and then they destroy it. And so the neutrophil, which is supposed to protect us, it's destroyed. So we are left armless. Enterotoxin heat stable. That's another toxin and resist hydrolysis by gastric and intestinal enzymes. So if the bacteria goes inside our intestine and our stomach, the gastric area, these enterotoxins, which are heat stable, they prevent the digestion of Staph aureus. Exfoliative toxins, a protease, which means it's what? It's an enzyme. And what does it do? Look, this is the bacteria on the surface. It goes down into this wall of tissue, cells, these are cells. The PMNs come to fight it, and then it is unable to fight it, and then this makes a big gap here. This is exfoliative toxin. It's a protease, it's an enzyme. TSST, toxic shock syndrome toxin. And this is a disease which I will show you how it looks in the next few slides. This is a super antigen. This is T helper cell in the immune system. It's the maestro of the T lymphocytes. And this is the antigen presenting cell. And the TSST, which is the green here, comes as a super antigen, big antigen. And this big antigen causes the release of lots of interleukins and cytokines. Usually the amount of interleukins and cytokines that are produced by just normal antigens, it's very small amount. But with this TSST, when it attaches, look, it attaches to the T helper and the antigen presenting cell APC, the interleukins and cytokines, they form a storm of interleukins and cytokines, which makes the inflammation very big and very severe. That's just a quick look about the enzymes. We already talked about this, but It doesn't harm if we just make it in another way. Look, catalase breaks down the peroxide H2O2 into water and oxygen. We'll talk about this in details in the lab. Staphylokinase digests blood. Hyaluronidase digests connective tissue. All of these are in the Staph aureus. Nuclease digests DNA, nucleic acids. Coagulase clots plasma. Penicillinase inactivates penicillin as the antibiotic. And lipase digests or breaks down lipids. So all of these 1, 2, 3, 4, 5, 6, 7 commonly known enzymes, that's what they do to the host tissue. And as again, I'm going to repeat this, that all of these enzymes, the human beings, microbiologists, doctors made use of them. Catalase for the catalase test. Coagulase for the coagulase test. Nuclease, there is a nuclease test also. Staphylokinase treatment. Let's go to the diseases. Staph aureus causes a series of diseases, group of diseases, and let's go to them one by one. We are not going to talk in details about these. This is SSS, which is Staphylococcus scalded skin syndrome. Scalded is like burning someone with hot liquid or steam, causing the peeling of the skin. Okay. Caused by exfoliative dermatitis, caused by toxins A and B, alpha, beta. So that's one. SSS. Then we have STSS, which is staphylococcal toxic shock syndrome. If you see the palm, this is the palm of our hand, the front of our hand, this is the palm. It's desquamation. Also, the skin is peeling. If you see somebody with his palm or her palm, skin is peeling, this is STSS. It's a staphylococcal infection. Folliculitis. This is the inflammation or infection in the follicle of the hair. Exactly. And that's why many ladies, many human beings, but particularly ladies, they have it and then they start squeezing it out until they damage their skin. But this is Staphylococcus aureus infection, S. aureus infection. Of course, it can happen from strep also and other bacteria, the Propionibacterium acnes. But also staph can cause it. Folliculitis, inflammation of the follicle of the hair. Also, we have impetigo, staphylococcal impetigo, which means there is also streptococcal impetigo. Impetigo is this production of vesicles like these that rupture and crust over without an E here. There's a crust cover. It dries out. There's this ulcer or small lesion or vesicle, and it ruptures. It dries out because it's exposed to air, and crust forms. Crust like a cover. Crust, formation of crust. This is staphylococcal impetigo. There is also streptococcal caused by Streptococcus pyogenes. Look at this, Staphylococcus furuncle. I used to see lots of these. Many soldiers and many patients used to come to the lab, and they want us to test for leishmaniasis. They thought this is leishmaniasis, but it's bacterial. This is what we call Staphylococcus furuncle. What's a furuncle? It's a boil. This is a boil. Skin abscess caused by Staph aureus and involves the hair follicle and the surrounding tissue. Folliculitis, only the hair follicle. This one, the hair follicle, and wider. The circumference, the circle is bigger. There is also the carbuncle. So this is furuncle, one, and there is the carbuncle on the elbow here. And this is here. This one is one furuncle. This is a group of furuncles. The carbuncle is a group of furuncles, and they are attached to each other from beneath. So clusters or group of furuncles with multiple pus heads, that's 1, 2, 3, connected subcutaneously. Under the skin, they have the same connection. But on the surface of the skin, they come as different furuncles, 2 or 3. This is we call carbuncle. Furuncle, carbuncle. Again, I'm just going to mention these diseases that are caused by Staph aureus. And as you see, Staph aureus, wherever it goes, it makes a problem. Although, as I said, in part of the people, they can be normal microbiota. So bacteremia, what do I mean by bacteremia? You have the staph in the blood, and we said that the blood is sterile. So anything goes inside the blood cause a problem. Even the staph epidermidis, which is normal microbiota and harmless. So any localized infection can become invasive and lead to bacteremia, bacteria in the blood. Endocarditis, inflammation of the endocardium, inner lining of the heart. Osteomyelitis, infection of the bone, osteum. Cerebritis, infection or inflammation of cerebrum in the brain. Pyelonephritis, which is kidney infection. UTI, but upper UTI. So this is a group. In addition to those which I showed you, which were all skin, these are systemic. In the blood, in the bone, in the brain. You just can't tell how dangerous Staph aureus can be. Other diseases also. S. epidermidis and S. saprophyticus. These are the ones which we are interested in. The saprophyticus, more dangerous than epidermidis. Urinary tract infections in sexually active females. Epidermidis, as I told you, epidermidis is harmless, doesn't do anything. However, in certain circumstances, it can be a problem. Example, if your immune system is very weak, then they are opportunistic. They come, yes, they stand up and they make problems. Also, catheters, prosthetic devices. You have devices in the elbow, prosthesis, prosthetics, like artificial parts inside our body. We call these prosthetic devices, and they can be in the hand, they can be in the elbow, they can be inside even. Pacemakers, maybe shunts inside, yes, to the heart. All of these, if they are contaminated and they have and go inside you, they cause a problem. Bacteremia in neonates in ICU. Neonates, the newborn. In the ICU, intensive care unit, S. epidermidis was shown to be a problematic. And saprophyticus, sexually active females. Schleiferi, Logdinensis, Hemolyticus, all of them are normal. But if you put them in the wrong place inside your body, then they become abnormal. That's the idea. Look, endocarditis, Schleiferi, endocarditis, septicemia, osteomyelitis in the bone, joint infection and wounds. Endocarditis also here, bacteremia, peritonitis, infection of the peritoneum, UTI, endocarditis, bacteremia, peritonitis. Laboratory diagnosis, specimen collection, specimen transportation, specimen processing, which is the pre-analytical or pre-test phase. We talked already about these in the chapter, which is the specimen management chapter. Diagnosis. As we said in the lab last week, that gram stain, it's the cornerstone of all tests. So first thing you have to make is the gram stain. You can make it, as we said, from the culture or from the clinical sample. And if you look here, they are purple or violet, which means they are gram positive. And they are cluster shapes, as it says, staph. Staph means cluster. So they're cluster shaped and gram positive cocci. Actually, it's cocci. Coccus means it's a ball-like. Under the microscope, it's 2 dimensions, circles. This is the Micrococcus luteus or Micrococcus species, tetrads. If you look here, they are in 4s mainly. And not only that, they are a little bit bigger than this. Gram stain, if you are experienced under the microscope, you will see they are tetrads, plus they are a little bit bigger. Diagnosis. Diagnosis here, direct detection method, nucleic acid detection, which is molecular, which means it's PCR, nucleic acid amplification test, NAT. And we have an example. Most molecular methods are based on targeting genes. Example, MIC-A gene for Staph aureus MRSA, methicillin-resistant Staph aureus, which is the antibiotic-resistant species or variant of the staph. Not all staph are MRSA. Staph usually, when you give it antibiotics, it's okay. One variant, which is this, it's very resistant to antibiotics. We call it MRSA, methicillin resistant. Some people, they call it MRSA. Some people call it MRSA, as you like. And usually they use this MIC-A gene to detect. And this is the gene, the way the gene is depicted. MIC-A and MIC-R. This is the one. MIC-A is encoding, this one, the yellow, the golden, encoding methicillin-resistant genes. Not all staph have this gene. Not all of them. The ones that have it, they become resistant. That's why PCR is very important. And that's the gel electrophoresis if you have ever made PCR. Have you made PCR before? Okay, this is the gel. Look, we have also commercial kits. Becton Dickinson here, Becton Dickinson Max, Staph, etc. There are, or you can just make it homemade in the lab. Just go for the MIC-A gene, the sequence, primers, and then you can just make it very simple. Fisher Scientific, that's another company, Becton Dickinson and Fisher Scientific. And others, of course, these are not the only ones. Nucleic acid detection. As I said, we go to MIC-A gene. And last time also, I told you that the 16S ribosomal RNA is the main one that microbiologists and researchers go for. But that doesn't mean these are the only ones, 16S or MIC-A gene for MRSA. No, there are other targets. And people and scientists have used them. Example are these. Of course, 16S and 23S, but 16S is more common and comes on the top of the list. Highly sensitive and highly specific. That's the PCR in general, not only this one. But there are also nuclease gene, NUC gene, ribosomal RNA, rRNA, which is 16S ribosomal RNA. Maybe this is for this. Elongation factor, tough. DNA gyrase, superoxide dismutase, so the A. GAP gene also. This also used very much. GAP gene. HISP60. HISP60. We use this, the HIP70 for leishmaniasis in parasitology. HISP70. Here we have HISP60, heat shock protein. And IS431, Staphylococcus insertion sequence. This list has been used as targets for PCR in the literature. Of course, as I told you, 16S was all the time the leading. Culture. This is conventional because many students, they can ask, why don't we use PCR and just finish off the culture business? Well, still people, they depend on conventional methods like gram stain and culture and even biochemical tests. We cannot say goodbye. It's too early to say this. Maybe 10 years from now, people will say goodbye to the biochemical test and depend mainly on PCR. At the moment, for many reasons, maybe commercial, maybe others, maybe scientific, still people depend on biochemical tests like indole and others, and catalase and coagulase. So general purpose media is the 5% SBA, sheep blood agar, chocolate, and also enrichment media, thioglycolate broth, and brain heart infusion. In my lab, we use TSB, trypticase soy broth. Also, we have what we call the selective media. It's like McConkey, but this is for staph. It's MSA. And many labs use this. It's MSA, mannitol salt agar. What is mannitol? Type of a sugar. This media has 7 to 10% NaCl, salt. Many bacteria, when you reach this concentration of salt, they die. They cannot tolerate. But the Staph aureus is salt tolerant. We call them halotolerant. That's one. Many bacteria will die if you reach 10%, but the Staph aureus remains alive. Plus, we have the mannitol. So this salt, what does it do? It makes it selective. Then we come to the differential. We go to the mannitol. Mannitol, phenol red. Staph aureus usually yellow. Differential, okay? So the original color of the media is mostly red. But if it's Staph aureus, fermentation of mannitol and becomes yellow. That's the idea. So original color is red and the new color yellow, from red to yellow. Okay, and this is commonly used in the labs, as I said. Many labs use this to make life easier for them. PAA, phenyl ethyl alcohol agar, inhibits gram-negative bacteria like, let's say, Escherichia coli and Klebsiella and enterobacterials in general. They are inhibited by this, and this just allows the gram-positive to grow, which means this is what? Is it differential? No, it's not differential because we don't have any indicators, or we don't have in general any change in color as a yeah. Colistin nalidixic acid. Colistin is an antibiotic. Nalidixic acid is also an antibiotic. So colistin antibiotic and nalidixic acid as antibiotic, both of them, they exist in the can agar and make can agar as selective. So the selectivity of the agar comes from salts, comes from antibiotics, sometimes comes from the stains, but the differentiality comes from changing color. So we have here. That's why people use very much MSA, but I have never seen people using the PAA or the can agar. If you look, this is the mannitol salt agar again. 10% salt kills all the gram-negative bacteria, making it selective. Phenol red pH indicator changes from red, the media from red to yellow again, becomes differential. And this is an example, S. aureus. Look, this is originally red, it became totally yellow, which means this is staph and it's aureus. Here we have some growth. See the growth here? It's staph, but not aureus. Here we don't have growth, nothing. So inhibited growth. No gram-negative, no gram-positive, nothing. So I think this is it for today. If you have any discussions, any questions, I can answer. Otherwise, see you next week.